Thank you very much for the introduction and also for the opportunity for me to present this work on Steatoda nobilis. So first of all, Steatoda is a spider genus containing more than 120 species. It's worldwide distributed, but we'll discuss on it later on. And what is very important is that it is the closest relative of the true black widows of the genus Latrodectus, and consequently known, Steatoda are consequently known, of course, as false widows spiders. If we focus now on Steatoda nobilis, you have to know that it's native from Madeira and Canary Islands. You have here the localization. And its first record in England was in the late 19th century. Then the presence of Steatoda nobilis was well established in the 80s. Then it expands rapidly across all the UK in the late 20th century. Then in the whole coastal Europe, in France, Belgium, Italy, Spain, and more globally in many parts of the world, like in California, in South America, or again in Turkey or in Iran. But going back on the situation in UK and Ireland, 5 species of Steatoda have been reported to live there in urban and rural areas, always associated with man-made structure, which makes the Steatoda nobilis a synanthropic species. And if we go even deeper, Steatoda nobilis is strongly invasive due to not only an easy adaptation to many biotopes, of course, but also because female has a high reproduction rate because she can produce up to 1 egg sac per month with an average of around 98 eggs per sac. And this production of eggs can occur all along the year. First point. A second point, Steatoda nobilis has also a very high life expectancy, up to 5 years, which is often better than endemic species. And if we put all together, we can say that Steatoda nobilis have the potential to outcompete native endemic spiders and should then get all our attention. Alors, Steatoda nobilis is invasive, okay, but what does she eat? She eats wasps, worms, and any kind of insect, which is, of course, very classical for a spider, but more interestingly, small insects are not the only target of such spiders, because she can attack, capture, and eat larger animals such as lizards here, pipistrelle bats, or again, shrews. But if the spider can attack such small mammals, what about human cohabitation? In this recent article, J.P. Dunbar and Michel Dugon demonstrated that a bite of Steatoda nobilis can induce Latrodectus-like symptoms like local, regional, and radiating pain, but also erythema and swelling. They also demonstrated that a small majority of bites are associated to female that are most of the time larger than males. Altogether, this story activated a wish to know more about this spider and its venom, and we decided to do it in 2 parts using 2 technologies. The first one is the imaging mass spectrometry, and the second one will be a more classical proteotranscriptomic approach. First of all, as a technological challenge, we wanted to have a look into the anatomy of male and female venoms by the means of mass spectrometry imaging. So how does it basically work? The spiders are initially sacrificed using a CO2 chamber, then incorporated into gelatin, as you can see here on the pictures. Once deep frozen, the spider is sliced on slices of 14 micrometers thickness, and part of the slices are sent for staining to make classical histology, or are deposited on ITO slides to make mass spectrometry imaging. There is example of stained slices for male here and female spiders, where we could already be able to identify some of the organs, like the venom glands you can see here or somewhere here. There is now a first example of a male spider embedded into a frozen gelatin and cut according to the frontal plan. You can easily detect the abdomen, the cephalothorax, the legs, and also already the venom glands that are clearly identified here on the picture. For the female, cut according to the sagittal plan here, it's a little bit different, as you can again localize the venom gland, of course, but also see that in the female abdomen, you have plenty of eggs, and then the female was about to give birth. After the slice are done, we had to deposit a matrix on the slice. A matrix is a small organic compound that allows the process of MALDI to occur. Then we define the resolution of our image. Here we have worked with 100 micrometer of lateral resolution, but we can go up to 20 micrometer. Then the acquisition is done using MALDI FT-ICR mass spectrometry. FT-ICR mass spectrometry allow to measure exact masses or very accurate masses, and also to record one spectrum on each position. At the end of the experiment, we obtain what we call an average spectrum, from which we can extract the M/Z of each ion and get directly the distribution of the molecule related to the ion on the slice. So to demonstrate the power of this technology, let's have a look into organ-specific metabolites. And to help you to appreciate the next result, this is an image of a classical anatomy for a spider with the different organs. I don't know if you can read, but I will in any case help you. So this is the first image. In this first image, you have the distribution of these three ions, one of it being already identified as a carnitine. As you see, the molecular density shown for this ion is clearly localized on a small part of the slice, indicating that these three species are specific to this organ. We believe in this case that this organ corresponds to the silk gland. The second image here shows the distribution for a particular lipid, a triglyceride here, 52. And as you can see, it's clearly specific to the outline of the abdomen of the spider, highlighting then that this lipid is specific to the abdomen cuticle of the spider. The third one is maybe easy to interpret because it's the signal given for two lipids. One is not identified because we have two isomers, but the second one is well identified. And the shape of this distribution here is in any case very typical, and this lipid seems to be, or are at least, specific to the ovaries or to the eggs in formation into the abdomen of the female. The fourth image here shows the distribution for another lipid, the sphingomyelin here, that is detected in two different localizations. In the middle here of the cephalothorax, it's easy to determine that this distribution corresponds to the brain, and this part is corresponding to the eyes. So this lipid, sphingomyelin, is a kind of marker, at least for the imaging here, of the nervous system of the spider. And finally, because we are toxinologists, more or less, at least for me, we have here a distribution for 4 ions. Some of them are lipids, and this last picture shows that these compounds are very intense and distributed in the cephalothorax of the spider in a large organ, which is, of course, here the venom gland. So we have shown here that the technology is powerful to map the metabolites into animal bodies, such as spiders, but what will be now our next step? we would like to identify the metabolite without any ambiguity, which is not done today because metabolites I showed you are just identified using their exact mass, but we like to have access to MSMS data to confirm, of course, their nature. And this can be made by nano-XC MSMS, either by total metabolomics, by extracting the total metabolome of the spider, or preceded by laser microdissection guided by MSI, imaging, to have, in this case, only the metabolome of a particular organ. And then we, of course, want to link this molecule to their biological role. Then, as the previous images were recorded in positive ionization mode, we would like to explore the negative ion mode to highlight even more metabolites families, and we are sure that this will work. And of course, we would also like to determine the interest of cutting according other slicing plans, frontal and transverse. I can show you here the first example of a male cut according to a frontal plane. So you have here the histologic slice, and you detect already the venom glands here, and some of the spider organs are also visible. If we extract the same signal than the previous one, we are totally able to identify again that the 3-acylglyceride here is specific to the abdomen cuticle, and that these 4 ions are also localized precisely in the venom glands. So that's it on this first part and about this technology, but of course there is still a lot of things to do with mass spectrometry imaging. The second aim of this presentation is to have a look into the content of male and female venom, and of course to compare them. For doing so, we started by an extraction of mRNA from the glands of male and female. Then we produce transcriptomics data, and we get more than 60,000 ORFs from this first experiment. If we compare the gene expression level of the 2 spiders, it appears that the female show a higher global expression of the gene corresponding to toxin and to enzyme compared to the male here on the right. And we can already see that the level of toxic gene coding for alpha-latrotoxin is very captivating. Moreover, female seems to express in proportion more toxin gene than enzyme gene compared to the male, who display a more equilibrated pattern. We can compare these results from transcriptomics to a classical 1D SDS page gel. You have on the left here the male venom and on the right the female. And even if the venoms have more or less the same mass range of protein in their composition, it's clear that female seems to produce larger proportion of toxins of high molecular range, which could be associated to alpha-latrotoxins. To validate and complete the study, we generated, of course, proteomics data using XC-MSMS system, including here a Q-exactive plus hybrid mass spectrometer, which is an Orbitrap. And the MS and MS spectra obtained were analyzed and combined 2 transcriptomic data using PixX+, which is a dedicated proteomics software, very powerful. Here are our results. For each venom, we obtained the sequencing of more than 250 proteins. And each protein have been, you have here the example of alpha-latro and sectotoxin, but each protein has been identified with high confidence due to the high number of peptides that can be that come to validate the sequence of the ORF. And this is validated not only by the exact mass, but also by, of course, the MSMS spectra we can extract for each of these species. This histogram shows you the similarity between male and female venoms. And what we can extract from this histogram is the most powerful toxin classes, alpha-latrotoxin, alpha-delta-latro insectotoxin, alpha-latro crustotoxin here, and all the enzymatic machinery allowing the venom to spread more easily are present in both venoms. If we now have a look into the differences between the venom, then the male venom contain more protease and enzyme isoforms than female. And in contrary, the female seem indeed to produce larger quantity and variety of the toxin. And these results are totally in line with what we have already seen from transcriptomics and from the SDS page results. Of course, a more quantitative approach is under preparation to describe even deeper these two venoms. In conclusion, Steatodonapilus is rapidly increasing its population density in a large part of Europe, and male and female venoms contain both high proportion of Latrodectus-like toxins, including alpha-galactotoxin, and also a large variety of enzymes responsible for Latrodectus-like symptoms. And of course, this study, I want to convince you, that Steatodonapilus should be considered as an invasive species with medical significance. Female venoms are more concentrated in toxins, and this allows them to feed more easily on large animals like lizards, small mammals, and this could also explain partially why female bites are more often reported by people. Concerning mass spectrometry imaging, it's an emergent technique, at least in toxinology, that is promised to a brilliant future because this can help to elucidate metabolic pathways, to perform also studies guided by MSI, like metabolomics, proteomics, lipidomics, whatever you want. And it can be also used for understanding the diffusion and the activity of toxin directly on tissue, for example, in the case of necrosis. And of course, all the ideas you have to exploit this technology are totally welcome. Finally, I would like to thank all these people, and especially Damien and Fernanda from Liège that provide me all the data of proteomics and also integrated data proteomics and transcriptomics. I would also like to warmly thank these people from Galway, John and Michelle, who invite me to be a part of Steatodonapilus story, because it was in the beginning their own topic of interest. And I would like to also thank you for your attention, and I will be happy to answer your questions.
Hey, Loic, there's a question online from Maria Economopoulou, so maybe you could start with that one.
Can you get any information from Maldi regarding the type of toxin, defensive versus predatory, based on location, density? No, not at this stage.
Can you repeat the question, Loic?
Yes, sure. Hi, Loic. Hi, Maria. Can you get any info from Maldi regarding the type of toxin, defensive versus predatory, based on location, intensity, or any other parameter? And the question is not at this stage. We wanted in the beginning to have an overview on the wall spider because, to be honest, it was just funny to have an image of the wall spider. But now, to be more serious, maybe, we want, of course, to go deeper into the venom gland and to increase the resolution to see maybe how the toxins are, of course, organized into the gland and how they can also maturate somehow. But concerning their defensive or predatory aim, no, we don't have any idea at the moment.
Thanks, Loic. Questions from the audience? There's one from Juan Calvete coming up.
Hi, Loic.
Hi, Juan.
Very nice, very interesting study. I not remember if you mentioned it, but I guess these are venoms that are that pull from different individuals. Do you have any idea of how many individuals and what's the individual variability? Because this can affect the total calculation of number of proteins or proteoforms that you have in the venom.
Yeah, this is true. We started with 50 species of each, 50 male and 50 females. So of course, it's correct. It may influence the results. And now John Dunbar is collecting male and female, and we will also try to evaluate the individual variation in terms of venom composition. And it's the next step. Yeah.
Then spare a little bit a pico-nothing amount of venom for doing a profile, so that it will allow you to have individual variability.
Yeah, good advice. Yeah,
okay.
Thank you.
Yeah.
Yeah, one more question.
Hi, Loic. Thank you very much for the talk, and it's very exciting. I was a master's student with Michelle in 2019, so I always have a very soft spot for Steatoda nobilis. Just out of interest, how have you guys been able to compare how the latrotoxins in Steatoda nobilis compare to in Latrodectus? Because I think that was one of the sort of arguments, is if they were paralogs or not. So just interested if you guys have checked that out yet. Thank you.
Yeah, it's a good question. No, we don't have any idea about the real biological activity of these latrotoxins compared to the one coming from Latrodectus. But it's something that Michel wants to do so, because now we have the total sequences of this latrotoxin. We need to produce them and test them on cell lines and something else to compare, of course, with the real ones coming from real widows, finally. Yeah, but we did not already do that.
Look, I think there's a genome of Steatoda out now, isn't there, from Jessica Garb? I think she's just completed that.
Yes, is it Parasteatoda or Steatoda? I don't remember exactly, but
yeah. Yeah, do the latrotoxins you found match up with what she found in the genome, which was, I recall, 16 or something ridiculous, 20 or something.
Yeah,
latrotoxins. Does your data match up with that, the genome?
No, not yet. No.
Okay.
No, we don't.
All right, one final question, then I think we need to move on.
Thanks. Hi. For the multi-imaging, is there like a minimum expression of the proteins to be captured, or like lower expressed protein? Are you able to detect those, or?
Yeah, to be totally honest and clear, what we detected here are metabolites, so very small molecules. Okay. If we want to go and image larger molecule like proteins, peptide is not an issue, but proteins, then we have to change the matrix, so we can then promote the emission, the desorption of proteins, but we are at least limited to a mass range up to 20 kilodaltons. We can not go higher because it's hard to extract the large protein from the tissue. But what we can do, and it's something in development, but hard to achieve, is to make a on-tissue digestion, to digest your proteins into small peptides, and then you identify your proteins based on the peptide sequences. It's something usually done for medical imaging mass spectrometry-based studies. But for the toxin, we have another limitation, which are the disulfide bonds, because we have to reduce them ideally if we want to have peptide, if we want to have much more peptide to map your toxin sequences.
Great, thank you, Loic. In the interest of time, I think we should move on. So could you join me in thanking Loic for a great talk?
Thank you.